Hello guys, welcome to my YouTube channel. In this video, we'll see the detail about oxygen dissociation curve and the factors affecting it. So, as we know that proteins are fundamental building blocks of our life, which have many functions like structural support, catalytic activities, and signal transduction. Proteins are made up of amino acids which are folded into 3D structures. The primary structure of protein is defined by its constituent amino acid sequence, while the secondary and tertiary structures contain hydrogen bonds, ionic interaction, and hydrophobic forces. Now let's see what is hemoglobin. So hemoglobin is a complex protein which is found in red blood cells and responsible for transportation of oxygen from the lungs to the body tissues. Its intricate structure and unique properties enable it to bind and release oxygen efficiently, ensuring adequate oxygenation throughout the body. Now let's see the oxygen binding characteristics of hemoglobin. So hemoglobin has the ability to efficiently bind and transport oxygen, which is very crucial for its physiological function. This binding is characterized by the oxygen dissociation curve, which depicts the relationship between the partial pressure of oxygen and the percentage of hemoglobin molecule which binds to oxygen. So as you can see here, this is a graph showing oxygen dissociation curve and it is sigmoidal in shape. It reflects the cooperating binding of oxygen to the different subunit of the hemoglobin molecule. The oxygen hemoglobin dissociation curve exhibits a distinctive sigmoidal shape, which reflects the cooperative binding of oxygen to the four subunit of the hemoglobin molecule. The Hill equation is mathematically model the cooperative binding, which relates the oxygen partial pressure to the fractional saturation of hemoglobin with oxygen. And the binding of oxygen to one subunit induces conformational changes that enhance the affinity of the remaining subunits, which amplify the oxygen binding capacity. And the hemoglobin's cooperative binding is an example of allosteric regulation, where the binding of one ligand, that is oxygen, affects the binding of subsequent ligand. Now, the Bohr effect and pH sensitivity. So, the Bohr effect describes the decreased affinity of hemoglobin for oxygen when the pH of the blood decreases, such as during increased CO2 levels or lactic acid builds up. So, the curve will shift towards the right. So pH sensitivity. The hemoglobin's structure and oxygen binding properties are highly sensitive to changes in pH. Acidic concentration, in which the H plus ion concentration increases, causes the conformational changes that reduces its affinity for oxygen, and the curve will again shift towards the right. Physiological significance. The Bohr effect allows hemoglobin to efficiently deliver oxygen to tissues with high metabolic activity and carbon dioxide production, where the pH is mostly acidic. Adaptive mechanism. This pH dependent oxygen binding is an important adaptive mechanism that optimizes oxygen supply to meet the varying metabolic demands for different tissues. Now, the binding of 2,3-bisphosphoglycerate, which is abbreviated as BPG. So, BPG is a crucial allosteric regulator of hemoglobin's oxygen binding affinity. These small organic molecules bind to a specific pocket within the hemoglobin structures that induces conformational changes that decrease the protein's affinity for hemoglobin. So first is allosteric modulation. So BPG binding to hemoglobin triggers a structural shift that favors the deoxy state, making it harder for oxygen to bind. Second, oxygen delivery. The BPG induced decrease in oxygen affinity allows hemoglobin to more efficiently release oxygen to metabolically active tissues with lower pH and higher CO2 levels. Physiological significance. This allosteric regulation by BPG is a crucial adaptation that optimizes oxygen delivery to meet the varying demands of different tissues within the body. Now let's see the regulation part. So regulation of hemoglobin oxygen affinity. First is oxygen partial pressure. So hemoglobin's oxygen binding affinity increases at lower oxygen partial pressures, which allow it to efficiently unload oxygen to tissues with high metabolic demands. Second is pH and carbon dioxide. So decreased pH and elevated carbon dioxide levels, as seen in metabolically active tissues, reduce hemoglobin's oxygen affinity, promoting the release of oxygen. Third is allosteric regulators. Molecules like BPG and protons bind to hemoglobin, inducing conformational changes that modulates its oxygen binding properties. Now, the physiological significance of hemoglobin functions. So, first is oxygen transport. Hemoglobin's ability to efficiently bind and release oxygen ensure that tissue throughout the body receive adequate oxygenation to support critical metabolic processes. Second, pH regulation. The Bohr's effect, where hemoglobin's oxygen affinity decreases in acidic concentration, which helps in delivery of oxygen to tissues with high metabolic activity and carbon dioxide production. Third, allosteric modulation. So, binding to allosteric regulators like BPG allows hemoglobin to dynamically adjust its oxygen binding properties to meet the varying metabolic demands of different tissues. Fourth, hypoxia response. Hemoglobin's adaptations, such as increased 2,3-BPG levels, help maintain oxygen delivery during hypoxic conditions. It means during uh, low oxygen concentration, so which ensures vital organ receive sufficient oxygen. So, lastly, the conclusion part and its clinical implication. So, first, clinical importance. Hemoglobin's unique function and regulatory mechanism are crucial for maintaining oxygen homeostasis and supporting vital physiological processes throughout the body. Second, diagnostic application. Understanding hemoglobin's oxygen binding characteristics and sensitivity to factors like pH and allosteric modulators inform the diagnosis and management of various medical conditions. Third, therapeutic potential. Insights into hemoglobin structure and regulation can guide the development of targeted therapies to address disorders related to oxygen delivery and utilization. Fourth, future research direction. Continued exploration of hemoglobin's complex biological properties and their clinical implication will drive advancement in undertaking and treating a wide range of health conditions.